Hi, we are students from Harmony School of Advancement. Our experiment was based on the bacterial conjugation, a process where one bacteria transfers genetic material to another. For our science principle, we would like to explain the medical advances that came about this process called bacterial conjugation, the spread of a bacteria gaining resistance to varying antibiotics. Thanks to this process, scientists were able to predict where the spread of resistance could occur to stop the problem of antimicrobial resistance. Through the knowledge gained, scientists can now not only predict the spread of resistance, but also figure out ways to neutralize that resistance, which in turn could improve healthcare practices.
For our social studies component, we talked about the history of bacterial conjugation and how genetic material was exchanged during this process. The concept of bacterial conjugation was first introduced by American microbiologist Joshua Lederberg in 1946. Lederberg's work focused on understanding the mechanisms of genetic recombination in the 1950s. Scientists like Francois Jacob and Elie Wohlman expanded on Lederberg's findings of plasmid genetic transfer. The identification and study of specific conjugative plasmids provided crucial insights into the spread of antibiotic resistance among bacteria, impacting both medical and agricultural practices.
For our English component, we wrote a journal that spanned over 4 days. On the first day, we learned about bacterial conjugation. We were then assigned a project about it, in which our group created a wrap and a visual model to engage the class. On day 2, we swabbed the E. coli strands, which contained either streptomycin or ampicillin, and let the bacteria incubate overnight to see if growth would be shown in the following day. On day 3, we observed the results. Bacteria had grown on the plates that were resistant to the antibiotic. Plates with no growth demonstrating no resistance. In order to conjugate the bacteria, we swab samples from each plate and applied them to a plate with both streptomycin and ampicillin, and repeated the incubation process. On the 4th day, we observed our results and concluded that the bacteria had successfully conjugated.
Here we analyze the varying results in between the strains and their resistance. In image 3, you see this plate contained both strep and ampicillin. Zone 1 had E. coli resistant to streptomycin, while Zone 2 had resistance to ampicillin. There was no growth seen since the plate contained both antibiotics. In our end result, image 5, we see that the strain of E. coli plasmid DNA was successfully transferred, allowing the strain to be resistant to both antibiotics.
In this graph below, anything above a 4 is positive, meaning it grew resistant. Meanwhile, anything before a 4 is negative.
This progress can create bacterial strains that are able to decompose organic waste, improving waste treatment and reducing environmental population, as well as holding a potential for advancements in biotechnology and medicine, which contribute to our civilization's medical progress.
The conducted lab allowed us to showcase how genetic material is transferred through a pilus, which leads us to spread characteristics of antibiotic resistance through this gene transferring method.
All right, guys.
Thanks
Thanks
for
for watching
watching
Harmony
Harmony
School
School
of
of
Advancement.
Advancement.
Bye.